Hello and welcome to our webinar Wednesday lineup. I am Helmut Brechel, Product Manager for Molecular Biology at Biosum. We are happy that so many people, customers and curious scientific workers join our webinar today. Thank you very much. The aim of Biosum webinars is to transport new and exciting information about laboratory technologies and new products to all interested people. Today, we would like to present an instrument for qPCR setup. MYRA, the first liquid handling system which can see. And we are pleased that Albert Weingart from Biomolecular Systems will introduce the exciting device and show us the easy and flexible setup. Albert is distributor and product manager at Biomolecular Systems with more than a decade of experience in qPCR applications. At the end of the session, we will have a few minutes for discussion. Please type your questions via chat into the system whenever you like. We will try to answer them at the end. Thanks in advance for your input. Albert, now please give us an insight into the MYRA.
Thank you, Helmut, for the introduction, and thank you for giving me the opportunity to present this webinar. On behalf of myself and BMS, I would like to say welcome to all attendees, and I'm very happy to have you here today. I would like to start by showing you an overview of this webinar. So the following topics will be addressed. First of all, who is Biomolecular Systems? I can imagine that for some people, the name Biomolecular Systems is new. So I'm going to explain who we are and where we come from. The next topic is why you should automate qPCR setup. After that, we are going to meet the MYRA. And I'm going to show you a setup of the MYRA and start a MIG run. After that, we will have a look at a video, short video, 3 minutes, in which John Corbett will go over the features of the MYRA. After the video, there is room for questions. So, Biomolecular Systems, who are we? Where do we come from? We are an Australian-based company, and we were established in 2010 by John Corbett and his father, and former Corbett Life Science senior personnel. We have an office in Sydney. That's where we do our R&D. We have a production plant at the Gold Coast, and we have a production plant near Brisbane. Looking at our history, we are not new to making instruments. We made the Robotic 2000, and shortly after that, we came out with the Robotic 3000. In 2006, the Robotic 6000 was launched. We also used to make robotic systems like this one. This is a CAS 1200. It's a PCR setup robot. And also the extractor gene, which could be used for DNA, RNA isolation. And today we have the MYRA. As Helmut already mentioned, it's the world's first liquid handling system that can see. Next to the MYRA, we also have the MIG, the world's first magnetic induction cycler for qPCR. And together they make a perfect team, and it allows us to create a seamless workflow because the MIG and MYRA are both driven by the same software. So a perfectly seamless workflow. What does it mean? Well, no more exporting or importing of sample names is required. Just set up, run, and analyze. But why should you automate qPCR setup? Well, there are different reasons for that. Better reproducibility is one of them, day to day, from lab to lab. Another good reason is it will reduce human error. Besides that, you can also reduce costs. by using smaller reaction volumes, and fewer mistakes mean fewer repeat experiments. Injurious for your staff. But last but not least, good qPCR reaction setup is the most critical part. The more experienced and careful you are in your pipetting, the better the real-time PCR results. So leaving all the pipetting up to a robot makes perfect sense. The key to excellent data starts with accuracy and precision. What do we mean by accuracy? Well, accuracy is the ability to achieve the right value, as can be seen here in this graph. So here we have low precision, low accuracy. Here we have high accuracy, but low precision. Precision is the ability to reproduce the same value. That's what we see here. High precision, but still low accuracy. For qPCR, you need to be able to do both. MYRA pipetting robot can do both, and the proof is in our results. I'm going to show you some of the results. More results can be found on our website, but if we talk about accuracy and precision, what we have done is we have made a twofold dilution. The dilution was made by the MYRA. We have 8 points, and we had series of 4 replicates. As you can see, the efficiency is 96%, and the R-square value is really close to 1. So let's push that a little. And what we did is 1.2-fold dilutions. And we can see that the accuracy and precision was maintained, even when we push it to the extreme. So the efficiency is still 96. R-square value is also close to 1. Some of the key features of the MYRA. All MYRAs come standard with an HEPA filter system, which is located in the back. Long life UV LED, which is used for decontamination, which is positioned just behind the pipetting head. And the MYRA also has a closed waste tub to reduce the risk of contamination, which can be found here. Accessories that come with the MYRA, the MIC rack cooling block that can hold the tubes for the MIC. It also comes with a multifunctional MYRA block, and this block can hold 10 ml bottles, 5 ml bottles, 2 ml bottles, various types of tubes. And that gives us the flexibility in setup without compromising space. As I said, the MYRA is designed to address the precision demands of quantitative PCR setup. Another nice thing of the MYRA is that it is very compact in size. It only weighs 10 kilos. And if you compare that to other instruments, it's less than half the size and weight. The MYRA offers true plug-and-play portability. What it means? No setup required, remove it from the box, and start pipetting. The MYRA is also highly versatile, and it is platform independent. So we can pipette in MIC tubes, but we can also do that in 96-well plates, 34-well plates, or 48-well plates. Can MYRA pipette in Robotic tubes? Yes, we can. The 0.2 ml tubes, but also 0.2 ml tubes, the gene disk 72, and the gene disk 100. Can I just use it for qPCR setup? No, you can also use it for pooling your samples, or cherry picking, or sample aliquoting. You can make serial dilutions, master mix aliquoting, DNA concentration normalization, and for setting up your NGS library quantification. The MYRA is also inexpensive to own and operate. Unlike other systems, we don't need expensive conductive tips. MYRA uses filter tips that can hold 1 to 50 microliter, and they are in a 384 well format. The reason why we don't need conductive tips is the fact that MYRA uses pressure sensing to detect the liquid level. Service contracts. No, we don't need those. They are not required. And why not? Well, the thing is that the MYRA comes with a removable pipetting head. So inside that pipetting head, it contains all the calibration data and the controller. So you can just swap and go. That means that there is zero downtime for calibration, so you just can continue. And there's no need for service engineers to come to your lab to do that. All MYRAs come with 2-year warranty. The standard warranty may be extended by an additional 1, 2, or 3 years to give 3, 4, or 5 years warranty in total. Easy to use. As I mentioned, it's the first pipetting robot that can see. So we have a camera, which is located at the back of the pipetting head. And what this camera allows us to do is it will make a single click plate calibration very easy. And I'm going to explain how this works. So select the plate you want to calibrate. In this case, we are going to calibrate this rack here. You open the calibration screen, and then you just point the crosshair over the middle of the tube, as can be seen here. Click on the green tick box, and you're done. That's how easy it is. So no more sticking your head under the hood and eyeballing the pipette tip positions. The MYRA does that all for you. It's really, really simple. Easy to use. The MYRA software. Setting up a MYRA run is very easy. You do it open the software, you're going to select assay from the menu option. You create your assay, and what an assay is, it holds all the information for doing the pipetting, including the cycling, but also the analysis setting. And I will show you that in a moment. Annotate your samples, set up the deck, and start the MYRA. You can start the MIC run directly from the MYRA software, and I will show you that also in a moment. It's very, very simple. So let's create a seamless workflow. I will switch to the software now. And here we have the software. And as I said, it starts with an assay. So we're going to make our first assay. And what we need to do is to tell what kind of chemistry are we using today. Intercalating dyes, hydrolysis probes. Let's say we want to do intercalating dyes, and we give our target a name, target A, and we can fill in a description here. And the next thing is that we're going to look at our reaction setup. So in our master mix, or in our reaction, we have water, we have source template, we have forward and reverse primers, and I'm missing one thing, that is our 2X cyber green mix. We can tell whether this is viscous. Yep, this one is viscous, and what it will do is it will just slow the pipetting, because this mix might contain glycerol. Do we want to have it mixed before use? Yes. The total volume is, let's say, 20. That means cyber green needs 10. And let's say for our reverse primer, we're going to use 2. And forward primer, going to use 2. As you can see, the water is automatically calculated. We're going to select the tube for our water, which is in this case a 5 ml bottle. The forward primer, that is a 2 ml tube. And our cyber green mix is also in a 2 ml tube. The next step is to define the profile, so our cycling profile. This is our profile, which is good, and we're going to add analysis. So after the run is finished, we are going to do a cycling analysis, and we are going to do a melt analysis. So this is it for the assay. You make an assay once, you don't have to worry about it again. We're going to save this assay, and we call it target A. Now we can start the run. And the only thing I need to do is to select the assay. In this case, target A. So the software already knows how I want to have my master mix made up. The next thing is that I'm going to annotate my samples. But before I do that, there is an option to choose which qPCR cycle you are going to use. In our case, we're going to use the MYC. The reaction plate we're going to use is a single MYC adapter, so 1 run. But you can also set it up for 2 runs and 4 runs. Annotating your samples is easy. We have 48, so let's do an autofill. So these are my 48 samples. The next thing is the deck layout. So here you see a graphical representation of the MYRA work deck. This is our target. This is the MYC rack, and we can move it anywhere we like. So what we want to have it there, yes. We have more than enough tips on board, so we're going to delete that plate. Yes. And here you have the multi-purpose loading block. The deck configuration, 96 wells. Yes, our samples are in a 96 well plate. This is our plate library. And I can choose from a lot of plates. Let's say that our plate is not there. Well, it's easy to configure a new plate. Just select the plate type, and you can select whether you have a cluster rack, or maybe a PCR plate, or maybe a deep well plate. So let's say that we have a deep well plate. We're going to give it a name. We're going to define the well shape, the capacity, and the height. So this is our plate. And now we have added that to our library, and we're going to add that plate to the work deck. And we can also do that with other racks. For instance, this rack here is a MYRA rack that can hold 36 well tubes of various sizes. Once we've done that, we move to the next tab, which is components, and we're going to add the components. Really easy. Here is our water. Here is our forward primer, reverse primer. We have our 2x cyber green mix, and we are going to make the master mix here. As you can see, if I hover over it, this is the amount of water that is needed to set up all those reactions. Forward primer. And this is where we're going to make our master mix. Simply right click, change tube, and maybe we want to have it made in a 1.5 ml freestanding tube. Okay. The last thing is to add our templates. And we can do that very easily with the filler like this. And we are ready to go. So the expected runtime is about 12 minutes from start to finish, setting up your master mix, making the master mix, aliquoting it, and adding your samples. I can imagine that you might not have a mic. You want to use a 96 well plate cycler, right? How does it work? Well, in a similar way. If we want to set up a PCR run for a 96 well plate, the simple thing is to just select the assay, target A, and in this case, we are going to use, for instance, a setup for a BioRed. And what we're going to set it up is in a low profile skirted plate. Yes, filling order needs to be horizontally, so it's filling it out horizontally. And we have 96 samples. Use the autofill, but you can no problem. The deck layout. So this is our 96 well plate. We're going to use the plate we made previously. And we're going to add our components. So here's your water, forward, reverse primer, 2x cyber green, and this is our master mix. That needs to be in a 2 ml tube. We're going to add the templates. Really easy. And this whole exercise just takes 23 minutes. So it takes 23 minutes to fill up a complete 96 well plate. Another example that I would like to show is how easy it is to set up gene expression. So just imagine that you have a MIC and you're doing a gene expression study and you want to look at 3 different genes of interest and 2 housekeeping genes. And you want to measure the gene expression for 3 different treatments. Now, for this, we need a total of 85 reactions, including a no template control. And this means that we need to set up the MYRA to load 2 MIC plates. So let's have a look at that. The only thing you need to do is to make separate assays for your genes of interest and for your housekeeping genes. And you're going to add these assays, because they contain all the information needed to do the setup. So in this case, we have added assays for my genes of interest, ACE, ACTN3, calcineurin, and these are my housekeeping genes. The samples, we've annotated them, and these reactions all need to be set up with ACE. These are my ACTN3 and so forth. And we have 3 different treatments: oxymetholone, testosterone, and nandrolone. The deck layout looks like this, where we have our samples and we have our reagents. So it will make 5 master mixes in total, and it will divide that over 2 mix plates, as you can see here. So these are my master mixes. These are my buffers, DNTP, and it will do that in 25 minutes. So after that, we want to analyze it. So let's have a look at that. Because the mix software allows us to combine those 2 runs and analyze them as one. That is what we call a project. So here we have 2 runs combined that we just made, A and B. The only thing we need to do is to analyze it by clicking on Gene Expression Analysis. And what we will get is the result in a graph, but also in a table. So here we have the 3 treatments, and this is our gene expression ratio. This is all done with the REST method, but we can easily change it to delta delta CT. You can see that this gene is no difference in result when treated with oxymetholone, whereas this one, for instance, ACE, is upregulated with testosterone. Yeah? Okay. Let's get back to the MYRA software, and let's actually start a MIC run. Going to add the assay once again. Select the samples. Deck layout. Our favorite plate. Components. And of course, you can make a template. You don't need to do this every time, all the time. Once you've made a template, it's very easy. Just load your samples and go. Yeah, change this tube to 1.5, and you're good to go. So after the setup is done, and I need to have my samples here, you can go straight into the mix software. Just click on this running man, and file will be saved. Yes, okay, thank you. Yes, save it here. Yeah. Successfully exported it, and it will open the MIC software for you. And I have a MIC connected to this laptop. So the only thing that I need to do now is to just start the run. The MIC is connecting. Let's start. And there it goes. And all the samples are now seamlessly transferred to the MIC software. So that is how easy it is to set up a MIC run using the MYRA. So let's move to the next topic and watch the video where John Colbert will tell you more about the MYRA. And after this video, there is time for some questions.
So we figured all these people with mix out there, they need a robot to set them up. So this is our new little robot we've called MYRA. It weighs 10 kilos, perfect for setting up qPCR. It's designed to work with the mic hand in hand. The software you run on the MYRA runs on the mic, so it's a seamless integration. 48 well tubes for the mic. They load straight onto the deck of the MYRA. We use 384 well tips. We've got a removable head, so you can easily take the head off. If you need to calibrate it, replace it, the head moves on and off in seconds. You can put all your master mixers and primers here, and you can configure this deck. And we think one of the major potentials is OEM kit manufacturers who are doing IVD. They can populate all of their tubes on the deck. The machine has a camera integrated into the head, so the robot knows if the deck's been populated correctly. It prepares all of your samples ready to load straight into the mic. So all sample names, etc. are seamlessly transferred. There's no ability to mess anything up. The two work hand in hand, and it's a complete turnkey semi-automated qPCR system. You can also drive multiple mics from the one robot, so on your tablet, and you can drive multiple mics, so you can keep punching samples through multiple qPCR machines to get your throughput up. And of course, once you've run all these samples in these independent mics, either in parallel or sequentially, you can then combine all of that data, analyze it as one big project. So we can analyze up to 480, I think, or 1,000 samples, all in the one project. So all of your data is unified, basically minimize any kind of errors. And that's about it. Thanks very much.
So that was it. There is now time to ask some questions.
So thank you very much, Albert, for this nice and interesting presentation. So we learned MYRA is really flexible and easy to program. In Germany and Austria, you can just contact us directly or our local sales account managers for further information and quotes. And now we are open for questions. You can type them into the chat box and we will answer. I think we will start with, do you need to calibrate the plates for every position?
No, you don't need to do that. Once you've calibrated, you don't have to calibrate it again. So it doesn't matter where you put your plates on the work deck. Once they are calibrated, you can put them anywhere you like. Yes, yeah.
Very good. Next one is, have you considered an 8-channel pipette?
Well, not really. Well, the thing with 8-channel pipettes is that they are difficult to calibrate, and what's more difficult is it's difficult to maintain all 8 channels to dispense identical volumes. So for precision, which is crucial in real-time PCR, the use of a single pipetting head is just a better option. And with the single pipette, we're also more flexible in our setup. So that's why we have a 1-channel pipette.
Very good. With a mic, you can control up to 10 mics from one computer. How about the MYRA?
Yes, you can control multiple MYRAs with one PC. You just add more MYRAs and there you go. You can do the same. Yes, you can do that.
Okay, very good. So I think that's it for today. So thank you very much again, Albert. our time is over, unfortunately. Other questions raised in the meantime will be answered by email. Thank you very much for attending the webinar. Have a nice day and goodbye.